Right. So there are some notes that I do want to make at the beginning of this because um where is it? Oh, there we go. Is if you have any conversations about human behavior, about mental health, about mental disease states, about poor, something. Usually, in our modern time, we start talking about things like neurochemicals. So, typically, when we talk about depression, we talk about things like serotonin. When we talk about different stimulant drugs, we talk about dopamine. Um, Opioid systems, what areas of the brain are associated with depressive symptoms, and once those are removed, does it take away a person's depression and those kinds of things? And it's not uncommon to kind of put life into making sense of the brain. But there are some cautionary tells that I do want to just express to all of you when you're reading material on the internet or on Facebook or Instagram about found a new way to rewire the brain and all of those kinds of things. Science isn't quite that far along in the field of neuroscience to be able to really measure that physical change or measure that rewiring, quote unquote. Um, the idea that depression is associated with serotonin isn't based on any measurable amount of serotonin in the brain. It's based upon a medication we give for it that actually only works for about 30% of patients. Um, and there's a growing argument, and I just want to bring this up to just kind of emphasize that while yes, we'll say some things with confidence in this class about the brain and all those kinds of things, I want to make sure everyone knows that this may change. And neuroscience and the study of the brain is really in its infancy. It really only really started in earnest in the 1980s. And it's almost younger than genetic science. Okay. And indeed, there's a lot of argument right now in the field among researchers about what is the efficacy of the research of looking at brain imaging and making associations with that because they use what's called linear math to analyze all of that. But the argument is that we should throw all of that research on brain imaging out because it's using the wrong kind of mathematical analysis. It should use the same type of analysis that we use for topography. It should use more geometry to analyze all these processes, not the linear statistical math that has been used on it. So even among the experts in this field, they're not even sure if they're even using the right type of analytical tools to analyze the data they get. So, and again, the reason why I bring this up is because this is a very common topic that the brain is everything and all of this, but we haven't even scratched the surface of even understanding it. And I just want to bring that up because in this section, it's going to look like we have this full conceptualized idea of what the brain is and how the brain operates. But I just want you to keep in mind that this is a very infantile understanding of the brain and its capacity. Okay. So I just wanted to give that pre-load before we get into this. But so what are some things that we are aware of? Okay. That the basic unit of communication within the nervous system, which the nervous system is a communication system, is the neuron. Okay. And we roughly have somewhere about 100 billion uh, in just our brain region, not including throughout the rest of the body. And if you want to think about this capacity of 100 billion um, uh, in the brain, I want you to think about if each one of those represented a byte in computer terms. That's 100 billion bits of information that our brain processes continually. But it doesn't start at 100 billion because each of these cells, these nerve cells, are connected to multiple other cells, creating other multiple abilities. Okay. And if we were to put this in concept in our modern age of AI, all right, I want you to think about this from this. So we're getting these data centers that require megawatts of electricity. They consume huge amounts of energy. They need constantly cooled and everything. But an AI system still does not have the capacity of a human brain. But all of that has to go into it in order for it to function. Okay, the reason why I bring this up is I want you to think how magnificent this little, I think it's like 4.5 grams in the top of our head. One time someone calculated how much it would take for a computer system to match one human brain. And the estimate is it would be the two-story building the size of Montana, completely filled with supercomputers, would only match one human brain. Now you're probably going, I don't know, humans, we're not that smart. We're not, but I want you to think about all of the other functions the brain does for us. It makes decisions quickly for us. And something that I will challenge you when we get to the learning and memory section is I will challenge you to go out to your car and make every single minute decision to get in your car and turn it on. And it should take you anywhere from 400 to 600 individual decisions, starting with which hand, where is it, which fingers, where's the keys, how am I going to pick up the keys, which fingers am I going to use to, and if you actually count the number of decision it takes just to get in your car and turn it on, it's somewhere between about 400 to 600 individual decisions, but your brain makes those decisions within a fraction of a second. Okay. And so it is a miraculous piece of machinery, and we should note that it is what that is, what it really is. Now, neurons, there's always been debate. Do we grow more neurons in older age and stuff like that? And yes, there is evidence that we do, but neurons are not a self um, um, restorative system. Okay. Once a neuron dies, nothing replaces it. Okay. Unlike every other cell in our body that regenerates itself every 4 to 7 years. Think about that. We get a brand new body except for our brain every 4 to 7 years. Kind of something to kind of wild to think about. But anyways, nerves do not regenerate. They do not, one just doesn't come in and one replaces it. So it has specialized cells called glial cells. These glial cells are kind of the handmaiden to the nerve. It comes and it cleans it, it provides it with nourishment, it provides it with support, it fixes anything that may have gotten damaged in some way. And so our nervous system is comprised of the neuron itself, but then it's kind of supporter neuron supporter cell. Okay. Glial cells will become important even in the notion of memory. We find that people who have severe memory decline as they age, it's directly associated with a reduction of glial cells in their brain system. So we will talk about that a little bit more again when we get to the learning and memory section. Okay. So I just want to play this video because it gives a kind of a good, really basic overview of the different nerve cells, and then we'll break it down a little bit more, and then we'll talk about how nerve cells communicate with neurotransmitters. So let's take a look at this, and then we'll go from there.
On my Instagram, biology and medicine videos, please make sure to subscribe, join the forum group. For the latest videos, please visit Facebook and Instagram. Please like, and here goes ask questions, answer questions, and post some interesting things, including our works. And you can also change the quality settings to the highest one for better graphics. In this neurology video,
Sorry, gang, I forgot to turn on the sound. We'll start over. Thank you for putting that in the chat. Hold on just a second.
terminal, where the impulse, Armando Surigan, biology and medicine videos, please make sure to subscribe, join the forum and group. For the latest videos, please visit Facebook, Armando Surigan. Please like, and here goes ask questions, answer questions, and post some interesting things, including our Armando Surigan, biology and medicine videos, please make sure to subscribe, join the forum and group. For the latest videos, please visit Facebook, Armando Surigan. Please like, and here goes ask questions, answer questions, and post some interesting things, including your artworks. And you can also change the quality settings to the highest one for better graphics. In this neurology video, we will look at the neuron. The neurons are the communication cells. They receive signals. What you see, smell, hear is thanks to a group of sensory neurons. Neurons also send out signals or information. When we move our hands every time we breathe, it's because of commands being sent via efferent neurons. So neurons are a big deal, and because of this, we have billions of neurons in our body. Here's a typical structure of a neuron. It consists of dendrites, which receive an information, a signal. The cell body, the soma, the axon hillock, the axon, where information or the signal in a form of an impulse is propagated through. There can be myelin, which wraps around the axon to help in insulation and speeding up the impulse. And all the impulse will end at the synaptic terminal, where the impulse, the information, is passed on to a target cell. So looking at it, an input signal is received by the dendrites, gets passed on to the cell body for integration, and then the output signal is released from the synaptic terminal to a target cell for a specific response, a desired effect response. The target cell in this case is another neuron, but this neuron is different in that it is an unmyelinated neuron. It has no myelin sheath wrapping around it. This means that the propagation of the impulse along the axon is much slower. When you have a neuron with myelin sheaths, the impulse travels much faster. But now you might be asking yourself, how is the information from one from a neuron is passed on to another cell. Well, let's zoom into this area here, where these two cells are close to each other, where they synapse with each other. Here we have part of the synaptic terminal of the first neuron and its presynaptic membrane. And here is part of the dendrite of the second neuron and its postsynaptic membrane. The gap between the first and second neuron is known as a synaptic cleft. In the ends of the synaptic terminal region, like here, we find many mitochondria and vesicles containing what's called neurotransmitters. These neurotransmitters are released for communication, for the communication process between cells. What happens is that when a signal arrives at the dendrites of the neuron, it will create an impulse that will carry this information and propagate it towards the terminal. This impulse is an action potential. The action potential will cause, once it arrives at the synaptic terminal, it will cause the vesicles here to release the neurotransmitters into the synaptic cleft, where the neurotransmitters will then bind onto the cell's postsynaptic membrane. So we can say that the synapse is the site for intracellular communication. And seeing that the postsynaptic membrane of the postsynaptic cell belongs to the dendrite of a neuron, it will receive this information and then create another action potential that will propagate along the axon towards the synaptic terminal. So a new input signal is received by the dendrites. It will be integrated in the soma, and then the information, the action potential, the information will pass along the axon towards the terminal, and will be passed on as an output signal via neurotransmitters that will target a particular cell. It can be a neuron again, or it can be a muscle cell or an endocrine cell, any kind of cell, depending on where the neuron is located and what its desired response wants to be, what effect it wants to cause. So now that we have an idea of how signals are being passed all around our body and how we receive signals all around our body, let's learn more about the soma of a neuron and how the neurotransmitters are packaged up, are made. So here we have a close-up of the soma of the neuron. We have the nucleus containing the genetic material, the rough endoplasmic reticulum around it with bound ribosomes and free ribosomes for protein synthesis. We have the Golgi apparatus for packaging, and we have the lysosome. Now the protein neurotransmitters are synthesized in the rough endoplasmic reticulum by ribosomes and then packaged up by the Golgi apparatus. So here we have the rough endoplasmic reticulum synthesizing new neurotransmitters that passes them onto the Golgi that will then package them up in vesicles. These vesicles containing neurotransmitters from the Golgi are then brought to the synaptic terminal. Here we have the synaptic bulb of the synaptic terminal that we're zooming into. So these vesicles containing the neurotransmitters and also mitochondria, they move down via microfilaments or microtubules, and they move to the terminal bulb here. The vesicles are in the synaptic bulb, where they can be released via exocytosis to the synaptic cleft when an action potential arrives. The neurotransmitters can be reabsorbed from the synaptic cleft and form vesicles, and then these vesicles can be recycled. They can travel back to the soma of the neuron where they were fused with lysosomes. The lysosomes will digest these vesicles for recycling. So neurons are a big deal, what we just talked about, for cellular communication, for sending out signals, and also receiving signals, receiving information. These neurons I just drew is actually a typical structure or typical shape of a specific type of neuron, an efferent somatic neuron, that is. However, there are a few types of structures neurons can be categorized into. And these structural categories, it can be determined by which part of the nervous system the neurons belong to, if that made any sense. I'll just draw a diagram to explain this. So the nervous system, remember, it can be divided into two major parts. That is the central nervous system and the peripheral nervous system. Now I drew two peripheral nervous systems because the peripheral nervous system consists of a sensory division and a motor division, or better yet, an efferent division. The sensory division of the peripheral nervous system consists of sensory neurons that look something like this. It has dendritic branches here, the axon on either side of the soma, the cell body, and then the synaptic terminal here. This type of neuron is categorized under the structure of a unipolar neuron. Now we also have another type of sensory neuron which is slightly similar in that it also consists of dendritic branches, but the dendrite will extend to the soma. Then we have the axon and finally the synaptic terminal. This type of neuron is categorized under the structure of a bipolar neuron. As you can see, the unipolar and bipolar neuron are only slightly different. The bipolar neuron, we have two separate processes separated by the cell body. So we have the dendrite and then the axon on one end. The central nervous system consists of many interneurons. Interneurons, you can say, are neurons that bring signals within the central nervous system, so from the brain to the spinal cord, for example. But we also have interneurons that act as glial cells, helper cells. Anyway, these interneurons have some completely different structures to other neurons. An example of an interneuron is this, what I'm drawing. And it doesn't look like it has an axon, but only has a cell body in the center with many dendritic branches around it. This type of neuron is categorized under the structure of an anaxonic neuron. Then we have a multipolar neuron type, which contains dendritic branches on one end and then straight away an axon terminal branch on the other, separated by the cell body. Some multipolar neurons in the central nervous system look slightly different than this, and we shall soon see what I am talking about. Now, the efferent division of the peripheral nervous system contains efferent neurons, motor neurons, which are all multipolar in structure. And this is the type of neuron I drew in the beginning of this video. We can have efferent neurons with myelin wrapping around it, or we can have efferent neurons without myelin wrapping around it. Either way, both are multipolar in structure, in that it consists of dendritic branches, and then we have the cell body, and then the axon and terminal branches. As you can see, this is the other type of multipolar neuron I was talking about in the central nervous system. The terminal branch doesn't have to be really close to the soma body. In the efferent division, the terminal branch is extended with the axon. Hope that made sense. And anyway, that was it for the neuron video for neurology. I hope you enjoyed it, and hopefully I'll make a video on an action potentials or action. Arman hazırlan.
All right, so I hope that was a helpful illustration of the uh, neuron as it is, and I'll just kind of go through these really quick. Again, he talked about the dendrites. It's what receives the information from another cell. The soma is kind of the central body. It houses the genetic material. It manufactures the neurotransmitters for that cell. And then the axon is what carries the action potential or that electrical impulse. Okay. Because what's going on in our brain is a chemical electrical event. The chemical is a neurotransmitter going into a cell. And if there's enough, it creates an action potential, which then creates electrical impulse down the axon of the cell. Okay. And then the axon terminal is then what was releases its neurotransmitter from the information it received from the others. Okay. And then the myelin sheath is a fatty layer that coats over the axon, which protects it, but also speeds up the transmission of the impulse. Okay, so if we look at it kind of and I just want to kind of highlight some of the components here to kind of give some better direction and some ideas of why this little cell in and of itself is important. So we're going to talk about two case scenarios, adolescence and then Alzheimer's disease. Okay. In 2013, somewhere around there, a group of neuroscientists found out that when a person goes through puberty, there is this 3 to 4 month period where the brain goes from almost completely unmyelinated to completely myelinated, as much myelin that our brain has. Okay. If you want to think about this, it's kind of like driving a car that can only go 25 miles per hour and then just within a short period of time can go over 1,000 miles per hour. Okay. In just a very short period of time. And this has started to change our perception of adolescence and what adolescents really are going through. Okay. Um, in that in that well, let me first explain why we think in our young childhood our brain is largely unmyelinated, and then when we get into our adult body through puberty it becomes myelinated. Okay. An unmyelinated brain is better at learning. It can, because it's processing information at a slower rate, it has more ability to connect information. So that's why, for example, the average child between the age of 2 to 6 will learn somewhere between 30 to 60,000 words. It's why if you have a toddler in the house, they're always asking why, because their brains are processing at such a slow rate. They have to be kind of repetition is really important for them. Okay. But then I want you to think about what it takes to be an adult. Okay. To survive in the world as an independent species of your species. Okay. It takes a much faster brain. So I want you to think about this in the guise of a child playing in a street. Okay. A lot of times when a child sees a speeding car coming towards them, they often freeze and they sit there and they're trying to decide what they should do. And that's what usually causes a lot of accidents with children. But as an adult, if we walk in the street and there's a car speeding away, we instantly react and get out of the way. And that's largely due to the brain being fast enough for us to make an action possible. Hence, the child will sit and freeze where the adult will act quickly, hopefully. So that's kind of the kind of evolution of why we think that the brain, when we get our adult body through puberty, the brain becomes myelinated. But what does this mean for the adolescent? Okay. Well, actually, before we get onto that, let me explain how the myelin sheath speeds up transmission. So, this is a myelinated cell. As you can see, it has these nodules on here. And use your imagination. This is an unmyelinated axon. Okay. And this is how the myelin sheath speeds up transmission. So in this unmyelinated thing, I want you to kind of look how long it takes to get down there. So the electrical impulse starts right here, and it has to see see how much time this is taking to get down the cell body. All right. But with a myelin sheath, it can jump from node to node. So the electrical impulse go da, 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 and done. So that's how it speeds up the electric impulse, is it allows the axon to skip between each nodule. Okay. And so this is what I mean going 25 miles per hour to going 1,000 miles per hour just that quickly. Okay. Going back to the adolescent example. So when mom and dad go to their adolescent and go, why did you do that? Maybe they made some stupid decision. Okay. And the adolescent goes, I don't know. Based on this evidence that we have now, they're telling the truth. They probably don't know because their brain is going so much faster than what they're used to in a very short period of time. A lot of times we think adolescents react and it looks impulsive, but it's probably because our brain is processing information much quicker than the adolescent is keeping up with it. Okay. And so what have we found is a major difference between adolescents who have a quote unquote easy adolescence, where they have an easy time with it, they don't have major traumas or anything, versus what we find with adolescents who have a difficult adolescence. Okay. We find that it really has to do with parenting figures and those disciplinaries around the adolescents. Adolescents who have an easy time through getting through those teen years tends to have a parent who responds like this. Why did you do that? I don't know. Well, let's sit down and start from the beginning and let's piece it all together. So you showed up at the school at this time. What happened next? And they sit down and they break down the scenario with the teen and work through the scenario with them. What is this doing? Is it's helping the child catch up with their brain and what it's calculating and what it's doing. It's kind of helping them train it and train them. Adolescents that tend to have a difficult time during adolescence have parents who do something like this. Why did you do that? I don't know. Yes, you do know. No, I don't. Well, until you can tell me what you did because you know what you did, I'm taking away your Xbox. You hear the difference in the ability for the adolescent to really understand this fast brain that they now have. One's given the opportunity to help break it down. The other one is just left to their own device, which can lead to continued impulsive bad decisions. So something to think about if you're a parent of a teen or know someone who is. Okay, the other thing that is important about this myelin sheath is when it comes to certain dementias. One subtype of dementia is Alzheimer's, which is a progressive disease dealing with memory loss and function loss over time. But what makes this disorder a progressive disorder? Well, what's attacking the cell is plaque. I'll do it with black. And what this plaque does, and if you've heard the commercials, they talk about it, amyloid plaque, and they're coming out with medicines that reduce the amount that's in the brain, which is very hopeful with people who are at the beginning phases of Alzheimer's. But what is this plaque doing? Well, what it's doing is it's attaching itself to the myelin sheath. Okay. And basically, if you want to think about it this way, what this does is it blocks the cell's ability to send an electrical impulse. It's kind of like suffocating it. Okay. So that plaque suffocates this axon And if there's no longer any communication, the cells will die because it's not being used anymore. There's no information being sent or transmitted, so it will not, that cell dies. Okay. Now the problem is if you get a couple of these cells that have plaque on them that are connecting to another cell over here, if this perfectly healthy cell is not receiving any information from these other cells that have this plaque on it that got suffocated, because this healthy cell is not receiving any information, it dies too. Okay. And this is what makes this process of Alzheimer's kind of a slow, progressive disorder. It usually starts with some mild symptoms, and then they get recognizable over time, because the plaque is slowly killing even healthy cells that it has attacked. So this is an example of Alzheimer's dementia. Now, I should mention that Alzheimer's is not the only type of dementia. In fact, it's not even the most common, but it is the most progressive, and it usually ends up being the most detrimental, to the individual. So something just kind of, when we get to memory, we'll talk about dementia, and we'll see that there's all kinds of different types, and Alzheimer's is just one type of it. But it shows what happens when the axon is damaged, even when it has this protective layer of myelin sheath. Okay. So those are two examples, one in adolescence and one in usually older age, dealing with dementia, of direct effect on a nerve cell can have detrimental effects or progressive effects, again, going from a slow brain to a fast brain, and really shows the importance of these individual cells. Okay. So in that video, they also noted um, the the transmission of neurotransmitters, and there's just some features that I want to indicate. Nerve cells do not actually touch each other when they're communicating. There's actually a little tiny gap between them. And what happens is when you have that electrical impulse of that nerve cell, if there's enough, it will release these things called neurotransmitter, which we'll go over here in just a minute. Each neurotransmitter is associated with some type of functioning. Okay. And so I just want to kind of make some notes about these neurotransmitters before we move on. I mentioned earlier that we really can't measure the amount of neurotransmitters you have in the brain. So how do we know that serotonin is associated with depression, and lower serotonin levels lead to depression? Well, we actually don't know that. That's kind of a myth that that is known knowledge. What we do is we have assumptions that that's what's going on, because the medication usually given for depression is what's called a serotonin reuptake inhibitor. Okay. You remember that the gentleman talked about how any unused neurotransmitters get reabsorbed into the cell and get recycled. Okay. Well, what antidepressants commonly do, these SSRIs, is they prevent the reabsorption of serotonin back up into the cell. So what it does is, because the neurotransmitter can't be reabsorbed, it then makes it so that there's a sufficient amount of serotonin within the neural gap to make a potential action in the next cell. Okay. So our assumptions that low levels of serotonin are associated with depression are because of a medication and what we think that medication does. but we don't have any direct evidence that that's one, what it does, and two, the reasons we experience depression from a biological process. So, and again, this example, I hope, shows you that we are really still in the infancy of understanding neurotransmitters and how they affect different mental states, for example, like depression. Okay. So, so what are some of the neurotransmitters that we will talk a lot about within this course? We will talk about acetylcholine, which is an excitatory neurotransmitter. It's associated with things like movement, function, learning, and memory. So it integrates, it's really active when we're integrating a memory, for example, of how to ride a bike. So those deal with learning and memory, and the acetylcholine then helps the brain transmit to the body which motor movements need to happen. Okay. Dopamine is one we will talk a lot about in this class because it deals with things like reward, planning behavior, motivation. It is the thing, well, I'll put it this way in this terms, and if you have a child around, close their ears or something. But the most amount, the highest amount of dopamines human beings release naturally is when an individual is having a orgasm. That's the most dopamine going in the body that we've measured, and so, but so there's that. But what about artificially? And we'll talk about methamphetamine. And if you want to know why methamphetamine is so addictive, it is suspected from at least mathematical models that a hit of methamphetamine quadruples the amount of dopamine that is naturally produced when you have an orgasm. And that's why a lot of people on methamphetamine can't have intimacy without having meth. They're unable to have it kind of in a natural way because the dopamine just does not meet that level that really they can feel anything. Okay. I just wanted to bring that up because that's dopamine. That is that chemical that is that reward for doing something, that's something that is a goal-driven something. Okay. GABA inhibits neurotransmitters that exert inhibitory control over the central nervous system, and it has some participation in mood. Mainly dealing, GABA really works with, for example, the heart and stomach systems to regulate things like heart rate and all of those things that also are part of our mood and our mood processes. Okay. And I'll just mention this really quick. GABA, if, for example, a lot of people who have high blood pressure or heart disease, they're put on a GABA inhibitor in order to slow the heart rate down because that GABA is making it so that the heart is racing too much. So just kind of a side note on that. Glutamate is another excitatory neurotransmitter. It tends to participate again. This is a large one in learning and memory. Norepinephrine is an excitatory neurotransmitter, is arousal visual. We'll talk about the sympathetic nervous system and its excitatory response when it gets you ready for fight and flight. And we'll talk a lot about norepinephrine and its effect on preparing us for the fight or flight and stress response. Serotonin is associated, again, we'll talk a lot about it when we get to mood and sleep, because the systems that tend to regulate those two things tend to have a large number of serotonin nerve cells. For example, I've been mentioning the amygdala over and over and over. It is very much associated with emotions, primarily fear. That's when it becomes most active, is when we're having a fear-inducing event. And then it releases serotonin to try and calm or understand this system that's going on. And then we have different type of neuropeptides. So for example, epinephrine release pain, endorphins release pituitary again to relieve pain. Placebos can release endorphin levels. These are really kind of the system. If you've ever known someone who maybe broke their leg, but they kept running, it's when they have so many endorphins in their system that the system ignores the pain of the broken leg in order for the person to survive. Okay. But we will also see its role when we get to stress and how much continued release of endorphin due to chronic stress can have a negative effect on the system as well. All right. I think that's a good place to stop because we got through the nerve cell, and then this coming Wednesday, we'll get through the brain system and the nervous system.